Press the record button.
And get us out.
I ask that people joining the talk to remain muted. And if you can, turn your camera off and we will allow for questions after the presentation. We'll give it a couple more minutes and then we'll get started. All right, it looks like we've had the majority of folks join us. And good evening. And thank you for joining us for our 2025 lecture series at White Mountain Research Center. I do have some housekeeping items to remind you of before we get started. First of all, my name is Gayleen Kinsey, and I am the scheduling coordinator at White Mountain Research Center, and I will be moderating this talk this evening. Some of our housekeeping items include, to keep your video off and remain muted until the Q&A. And before we begin, I would like to give a land acknowledgement. We do this at all of our events. The White Mountain Research Center acknowledges the Numu, Paiute Inuit, Shoshone as the first peoples and traditional land stewards of Paihunadu and Kohotoya, the Owens Valley and White Mountains. As a facility managed by a land-grant institution, we pay our respects to the elders and our relatives, relations, and friends of past, present, and emerging generations. And with that being said, I would like to introduce this evening's group. This is the largest group we've had present at a virtual talk. So here's a first, and they happen to be running one of the longest studies at White Mountain Research Center. It's been over 20 years now. I would like to introduce the speakers of this talk. First is Dr. Brian Smithers, Assistant Professor, Montana State University, GLORIA Great Basin Director, and former WMRC mini grant recipient several years. Jim Bishop, GLORIA Great Basin Safety Officer and unofficial historian. Jan Nachlinger, GLORIA Great Basin lead botanist. Dr. Seema Shelth, Associate Professor, North Carolina State University, Raleigh, GLORIA Great Basin Science Director. Kaleb Goff, PhD candidate, we'll just say this, NC State University, Raleigh, in Seema's lab, GLORIA Great Basin Field Coordinator, and also a WMRC mini grant recipient several times over. And the topic of their presentation this evening is GLORIA and the Whites, 20 years of long-term alpine plant community monitoring with community scientists. Here they will be discussing their nonprofit's long-term research that monitors the composition and distribution of alpine flora in a changing climate. And I will go ahead and ask Jim Bishop to unmute himself. He is this evening's first presenter. And with further ado, I'd like to pass the helm on to you, Jim. Or actually, it's Brian. Brian's the first one. I apologize.
Thank you, Gayleen, for that introduction. It's really, and for Gayleen, thank you for all of the work you do for White Mountain Research Center and for GLORIA. You're really a gem to have. So thank you. And thank you all for being here. I took a peek at the participants who are on this call, and it's a real who's who of the White Mountains, and it's just great to see some names on there, friends old and new. So thanks for being here. We've got I was kind of sort of making a joke earlier before y'all were on. You know, we've got 1, 2, 3, 4, 5 of us on here to speak, you know, when probably only one of us should be here. But it's sort of how we do things, which is get a whole bunch of people together and have a really good time while doing, I think, some really important science. So I'm just going to get us started off here and just kind of give the a bit of the preamble of how we got here. But Jim, actually, next slide, please. I do want to just mention that we are all part of GLORIA Great Basin, which is a nonprofit organization.
Not changing.
Uh-oh.
It just went.
Oh, there we go. Okay. Sorry. So part of we operate a nonprofit organization called GLORIA Great Basin that supports this research that you're going to hear about today. If at any point you get tired of listening to me and just want to look at our website or listening to any of our speakers, that QR code right there will take you to our website, which has got lots of fun information. Next slide, Jim. So GLORIA was born out of recognition of a couple of things. First of all, a recognition that we are really in a major biodiversity crisis. Biodiversity globally has been decreasing rapidly. It's pretty widely recognized that we're now part of the 6th mass extinction on the Earth, coupled with the fact that mountains really are these hotspots of biodiversity. They've got, they are centers of many endemic species that are only found in certain areas. And their native plants are really skewed towards these mountainous areas. A study had shown in the EU that alpine environments in Europe, which cover only 3% of the continent, make up more than 20% of all the native plants. And interestingly, despite the fact that biodiversity is decreasing globally, there was some evidence to support that in mountainous areas, biodiversity appeared to be increasing in certain studies. This is an older study on purpose by a collaborator of ours, Harry Pauli, from 2001, showing, back from 1800 until more modern, up to about 1992, potentially some increases in species richness, or the number of species that we see in mountainous areas. Next slide, Jim. And this appeared to be pretty consistent among a number of different areas. This is what we're looking at here are just some areas in the upper left there. The X is showing an historic condition of the number of species going back to the early 1900s. The diamond is the 1980s, and that circle is 2003. And so for each of these sites, it showed that there were an increase in species. The bottom right is showing 30 high summits of the eastern Alps. Same deal. This is showing the change in species number, or the change in richness. So there seemed to be a lot of evidence to support that that mountain areas were actually increasing the number of species. Go ahead, Jim. Interestingly, some studies were also reporting decreases in the number of species. And in fact, my home in the Rockies, in the Northern Rockies, was one of those places. So it wasn't all clear if this was actually a global phenomenon or if this was really happening. Another really interesting piece was that it didn't appear to be consistent across even the mountainous area. That these are showing for kind of 4 regions on a mountainside. We've got alpine, which is really that I think that kind of refers to their sort of subalpine forest down to montane forest, the alpine area down to tree line, high alpine, which is above that elevationally, and then the nivel zone, which is the very, sort of the top, or where the area that's covered in snow. And you can see that only the high alpine and the alpine areas appeared to be increasing in the number of species that were there. In fact, in the nivel zone, or that top snow-covered zone, mostly snow-covered zone, there were some decreases in this. Sorry, I said richness. In this case, they're talking about cover, which is how much of the land is covered by vegetation and material. So, it's a little unclear. We can go to the next slide. Here's just some of the species they were looking at in Europe that were showing some of these increases and decreases. If they're outlined in red, these are species. These are examples of declining species from the nivel zone. It's an Androsace on the upper left there. In the middle top, that's a ranunculus, and then a grass called Poalaxa in the upper right. A couple of other grasses, or another grass on the left, or a carex, sorry. On the bottom right, the two images are two species from a little bit lower down, the high alpine area. There's a Poa there and a Silene that were increasing in cover. Next slide, Jim. Meanwhile, we're also seeing across many different mountainous areas increases in tree line, or tree line moving up slope. And this was sort of interesting. It's potentially taking away alpine habitat. Definitely an indicator of some changes. But in Europe, it's tricky because humans have been using this landscape for many thousands of years. And so, are these trees moving up because of climate? Are they moving up because of the cessation of grazing? It's a little unclear. Next slide. So because of all of this, finally we get to kind of where we're going. A group led by a visionary by the name of Harold Pauli from in Vienna, decided what we need is this protocol to monitor these mountains, so that we are doing something consistent globally to look at these trends and see, like, are these things we think we're seeing, are they happening? And so we came up with this really nifty protocol, which you're kind of looking at diagrammatically on the right. Jim might go into a little more detail on this. So I'm just going to kind of mention that essentially it means you go to an area, you find 4 peaks that have the same substrate and then follow kind of an elevation gradient from higher to lower. And then you put a series of plots on there. You can see the photo is a group in Europe putting one of these 1-by-1 plots that we sample to try to and then we'll repeat these samples every 5 years, go back to the same places, try to put that plot down in the exact same spot. And we do a number of different data methods, and we collect a number of different data types. And the idea is that we will keep doing this for as long as it takes, or forever, who knows, but every 5 years we're going to keep going back to that mountain one way or the other. And so we've done that. This was launched in 2001 in Europe, and then we can go to the next slide, Jim. It has spread, and it's quite global. And I apologize, there's an updated image of this which I thought I'd put in here, but this gives you the idea that there are now GLORIA sites globally. It's found on 6 continents, polar to tropical, high deserts, Mediterranean climates, and was brought to North America relatively early in 2004. Connie Millar, I believe, is on this call, and she was the one to do it. And she instituted the first GLORIA plots in North America on Dunderberg Peak, where we're going this summer, and in the White Mountains, where we'll also be this summer. Glacier National Park also started, I believe, in the same year. And so it's this wonderful network of where everybody's collecting the data in the same way with the same protocol, in the hopes that we can really look at some global trends. And so next slide, Jim. I'll try to wrap this up. I'm going long. I said I'd go short. So here, these are our sites where we are at as an organization. We have two what we call target regions, which means we're monitoring 8 peaks in the White Mountains, and we've been doing it for a good long time as part of this really great relationship that we've had with GLORIA, with the White Mountain Research Center. I feel like we've both the organization, the GLORIA Great Basin organization, and the White Mountains have really grown up together, to this level, and so it's really great to be on this talk. And with that, I'm going to turn it over to Jim.
Thank you, Brian. Really nice overview and explanation of how we became involved in this, and that it is a global effort. I want to focus and highlight our history with the White Mountain Research Station or Center. And I want to emphasize that we certainly have benefited in many ways, and I'll highlight some of those, from working with White Mountain Research Center, but also many of the things we've been able to accomplish and practice and learn from in the White Mountains have also benefited us in our other endeavors, which are 6 other target regions throughout the Great Basin. And so it hasn't been just the White Mountains that have been benefited by our relationship, but throughout the work that we do. So I'll just remind you quickly, the alpine zone, as delineated here, is the zone above upper tree line. And it, you can see, is a very, very different landscape and ecosystem. That boundary is defined essentially by an isotherm that depends upon mean temperatures throughout the growing season. And so you can think about how climate sensitive that boundary must be, and therefore how climate sensitive the landscapes both above and below it might be. So we think about what kinds of changes might occur. Above that, it's colder. Below that, it's warmer. And so the alpine plants have managed to survive in cooler environments than everything below them, but it's as if they're ready to charge up given the opportunity. So think about some of the possibilities. Certainly warmer climate could change and shift that boundary and change the habitats both above and below it, but not just temperatures. Precipitation can certainly change in amount and timing. And another very important aspect of it is snow cover, which is critical to protecting alpine plants during the worst of the winter minimum temperatures, may be altered as well. And certainly those changes could cause a climate-sensitive alpine ecosystem to show changes, and it's our objective to monitor in ways that we could find that. So how do we go about monitoring it? Brian has mentioned the overview of summit surveys, and we'll go into a bit more detail with that. First of all, if you want to understand what's going on in the alpine zone, your survey sites have to span that zone from bottom to top. The critical thing that defines the ecosystem are the species that grow there, in our case, the plants, and some, not just which ones by identity, but some measure of the degree to which they occupy that space. So in the White Mountains, we have 2 groups of summits called target regions. The first group we did is on silicate substrates, meaning rocks that are dominated by silicate minerals. 3 of our peaks are granitic, and one of them is a combination of things, but the other prominent silicate substrate is quartzite. And so we have chosen places that range from just above tree line to the highest peak, which is White Mountain itself. This is a picture of our first day out on Sagehand Flat looking for a peak. This didn't turn out to be the one. The hard part was finding something that was 20 meters above the general landscape there, which we finally did. And this is just kind of a pictorial tour of some of the peaks in upper left. See if I can find my really cool marker. There it is. That's Sagehand Flat Peak. And this is actually one of the people from GLORIA International that handled the data that came out and joined us one time. This is Barcroft Summit, and this is White Mountain itself. Sometimes getting up White Mountain is a little challenging. This people powered the truck up the hill. And this is RNA Peak, which is directly east of Barcroft Laboratory in the Research Natural area. Our second group is on carbonate substrates, primarily dolostone, which is a calcium magnesium carbonate. And interestingly, you can capture all 3 of those summits in one photograph. Here, Sheep Mountain East on the left-hand side, Cottonwood South, and Patriarch Grove south. And if you could see down into the basin that's surrounded by these three peaks, you'd be looking down into Patriarch Grove in the White Mountains. The survey in general starts at the high summit point, or HSP. It's an altitude and azimuth controlled survey layout designed to be very simple so that people could apply it throughout the world without having any sophisticated means. So let's imagine we're looking at the south side or south aspect of this hill. Then this line would run down the southeast direction, this one down the southwest direction. Then we put a perimeter 5 meters below the surface, the peak in elevation, and one 10 meters below the peak in elevation. And that defines our largest plots. These plots here called summit area sections, which can be hundreds of square meters in extent. In the center, we have a 10 by 10 grid that is sampled at 400 points. And the most detailed survey is the 3 by 3 meter survey square in the middle, the outer corners of which are surveyed in detail as 1 square meter quadrats. So that's the general layout. There'll be one on each compass aspect of the peak. And sometimes it looks good on paper and it's really hard to string it out, but we do our best. And then you do the surveys, which is close to the ground work to see those tiny plants and properly identify them. A real focus of our work is on the 3 by 3s because they contain 4 1 by 1 meter quadrats and are surveyed intensively. Sometimes, as you see down here, even laying out the 3 by 3s is less than ideal. But people work, for instance, surveying here, the 1 by 1 meter quadrat in detail is every species that's visible there, and a gridded system of assessing the coverage. We also photograph all of our 1 by 1s and all of the boundaries and intersection points in the entire survey system so that they can be replaced on the land as they were in a previous survey. This is just an example of where we've photographed the 1 by 1 and labeled it here as to where it is. This happens to be Barcroft on the 18th of August in 2004. That was our first survey on Mount Barcroft. We're thinking that certainly climate, one aspect of climate that very well may control any effects on changing the alpine ecosystem is temperature. So we really need to have a way of measuring what temperature has actually done there. So we bury temperature loggers in the ground 10 centimeters deep, which is about 4 inches. And if you look at the temperature record of a sensor on the surface, you'll see, of course, the daily ups and downs like this. And at the 10 centimeter depth, you see a simple pattern reflecting the daily highs and lows, average about the same, but a much more muted lower amplitude variation in temperature from low to high. The other interesting thing you get out of temperature is, as down here, for several days, the temperature remained 0 degrees C, which is freezing, and that represents snow cover that remained 24 hours a day and kept the temperature 0 at the bottom of the snowpack. So you get not only temperatures and averages and amplitudes, but you get some measure of the duration of snow cover. We put these in a hole, take a picture of them so you can see where it is, and take another picture as a look when you approach the area, trying to dig it back up. And most of the time we find them. Once in a while, the ground-dwelling creatures find them first and dig them up. We collect plant specimens on each summit, and those are compiled and compared with herbarium specimens to make sure our plant IDs are correct. And our chief botanist, Jan, has made sure we've refined our identifications so we have a really excellent and clean list of properly identified plants. So when you're done, you have a view at several scales from a single square meter to hundreds of square meters in nested plots on different aspects, northeast, south, and west. Lists of all the plant species and different levels of accuracy, but measures of their cover. And photos, which allow us to resurvey the same plots and note any changes in the plants and the cover. Detailed temperature observations, and with that, measures of snow cover from which to gauge potential change or actual change. Well, that's okay on paper, but when we started doing it, we discovered there were some limits imposed by the fact that alpine plants are usually very small. They can be quite inconspicuous, and they often are present as a small percentage of the area, often a fraction of 1%. So while we had really good lists of the species, the estimates of cover were subject to lots of variability. The accuracy level was quite low. So we needed to consider how we can improve the accuracy of those, and we took this on in the White Mountains. We went one day in 2006 to a ridge near Patriarch Grove, and we spent the day assessing the accuracy and the usability of several different plot types and methodologies to better improve our cover estimates. And we came up with what ultimately was called the California method, or the 10 by 10 meter plots, and they lie in the center of each aspect of the plot system and provide much more detailed and quantitative data. Each 10 by 10 is divided into 10 meter long, 1 meter wide lanes or belt transects within which we identify every species. And then we survey with 40 points on a half-meter grid spacing to see what the, to identify the cover and the plant type and the substrate. Well, if that was going to be part of GLORIA, we had to convince GLORIA that was a good idea. So in 2007, we were joined by Hari Pauli on the right, who Brian has mentioned earlier, the science director for GLORIA, and our then field leader, Ann Dennis, who was really quite good with sampling methodology. And we stood up on Barcroft Peak and said, well, how about this and how about that? And Hari agreed that that was perhaps a good improvement. They went home and decided that we had begun with a 100-point survey, and he thought we'd be better off with 400 points on a half-meter grid. So we made that adjustment to the system. And it's now part of the International GLORIA protocols that you can use, and we use it on all of our sites in GLORIA Great Basin. So it's the mountains after all, and some days you start out with beautiful clouds and they get a little more troublesome as the day goes on, and you can end up with quite a downpour, which is not something you want to be out there in the field doing. Even if you don't mind getting wet, you don't want to get struck by lightning. So one of the great benefits of working with the White Mountain Research Center is having Crooked Creek at our disposal. And so on a day when you're not going to get back out in the field, at least you don't have to get wet. There's laboratory space, and you can continue with your work here doing plant identification in a warm, dry place with microscopes and many resources and references available. Or you can use all the space in the dining hall with its long tables to lay out equipment and sort it and organize it and get field sheets ready. It's a great benefit to have just the logistical support of Crooked Creek Station. But more than that, it's also a great place to eat out of the rain. And one of the wonderful things about working there is the interactions you get into, not just with the GLORIA gang, but with others who are there as well for different reasons. This happens to be one snapshot at one dinner. The gentleman who's gesturing is a glacier geologist. Behind him is a soil scientist. To his right in blue is our founder, Connie Millar. To his left is Daniel Pritchett, who is a White Mountain Research Center former, now retired staff member. And this lady right here is Ann Dennis, our field leader, and others. So you had quite a mix of people and wonderful opportunities to not just enjoy conversation, but to share information and get all the synergistic kinds of inputs that help all of us in our work in the White Mountains. I'll say one more thing. This also carries on into the evening in the upstairs meeting room, where we frequently have people with expertise in given areas share their expertise in these comfortable and laid back evening sessions where the presentations were really quite good and the discussions afterwards were good as well. So we had some ideas about wanting to see a little bit more about what's down the hill. The GLORIA Summit surveys take in the top 10 meters in elevation, and they're very intense there, and they do that on purpose. The summits are thought to be somewhat more pure climatically and not so much influenced by local terrain as areas further down. But there are reasons to think about what's below there. And so in our organizational meeting in 2004, we talked some about that, realized we couldn't get that started now. We had plenty to do just to get the whole GLORIA program going. But we wanted to do something that would reveal the species below the summits, give us a little better elevational resolution. For example, now between Barcroft and White Mountain Peak, the two highest summits we have is about 1,000-foot difference in elevation. Our downslope surveys, as we'll describe, have resolution of about 80 feet. So you get a dozen or so data points between those two summits. We want to be able to assess the plant distribution, the substrate distribution, and if you can develop a profile, so to speak, of the distribution of, for instance, plants, you can actually quantify with cross-correlation techniques any elevation shift that affects the entire population. So we started our prototype surveys in 07 and 08 using the then current 10 by 10 protocol and elongating it into 100-meter long belt transect. We improved that methodology in 11, 12, and 13 and have used it ever since. And so the downslope survey data has been useful, and Seema can highlight some of the things in ways in which it's been used to develop scientific output. We have a downslope survey on White Mountain Peak, and here in the diagram, each yellow line represents a 100-meter long on-contour, 1-meter wide belt transect. There are 14 there on Barcroft, 14 similarly on Sheep Mountain East, and it carries on down into the Limber Pine Bristlecone woodland, and on Patriarch Grove South, it starts barely in the alpine zone and goes extensively down into the woodlands. On Campedo Peak, which is not a GLORIA peak, we have transects that go all the way down into the shrubland below. So we have sampled an extended elevational range and quite a variety of habitats with our downslope surveys. A piece of the downslope survey, this represents part of a transect that would extend 100 meters along this contour. We break it into 10 segments in which we list all species present and take 40 data points to assess the degree of cover of each species in the substrate. Using our handy-dandy 2-point machine with a 0.5 meter wide points for the sample points, which keeps you from bending over quite so much. We do photo documentation at the ends of the transects and center so we can replace it where it was placed initially. And we take pictures along each 10-meter transect segment in order to illustrate the substrate and the plants. So another important thing that has been done in conjunction with the White Mountain Research Center is to develop a GLORIA Global Master Site, which are a series of projects that are relevant to the alpine zone, which are done by other researchers, and they extend over the entire alpine zone, include insect surveys in the butterfly count, plant recruitment surveys and microclimate surveys, and extensive studies of the demography of bristlecones and limber pines, paraglacial processes and soil formation, illustrated here, some of it, an insect inventory. Actually, that's 2 of our White Mountain Research Station compadres, Jeff and Yuta, that do insect surveys all the time. And the 1-day blitz intense butterfly counting session, which had a whole team of people come up and see all the butterflies they could find in 1 day and where they were and see how that changed over time. One of our colleagues from Palo Alto set temperature sensors out. This is looking down into the Crooked Creek drainage. This is Crooked Creek Station right here. And the red dots are temperature sensors from which he developed models that illustrate the temperature distribution within the topography, here showing the minimum temperatures that you'd get at night. One of the other things we did in 2008 was sponsored by the White Mountain Research then Station, now Center, was the CREC, we called it, Climate, Ecosystems, and Resources of Eastern California, which we put on in Bishop, hosted several days of talks and discussions and workshops, which were really valuable data sharing and information sharing events that coincided with the early years of our getting the GLORIA program underway. So against that background of beautiful alpine plants, I'll just mention 2 or 3 other things that are not directly related to the surveys, but which have grown out of our association with the White Mountain Research Center and which are ongoing. One is, you know, in the good old days, you stood on top of the hill, and if you'd never been there, somebody said, okay, do this and do that, take that string over there, and please bring me that. And it was okay, a lot of on-the-job learning, but we have developed training sessions. The first one was done out in the yard, so to speak, at Crooked Creek, that are much more thorough in helping people get oriented to the methodology so they can go up there and get right to work. It is our base for what we call our Peak Opportunity Fellowships, which is where we invite several students to join us for the week in the White Mountains and Crooked Creek. And we look to the White Mountain Research Center as support and inspiration for continuing surveys and research. And I'll end it with a picture of what is the center of our White Mountain universe, and that's Crooked Creek Station in the sunset. Thank you.
Thank you, Jim. So we're going to go ahead and have a short intermission so then Seema can load her presentation. There we go.
1940. I don't know how I get back to that thing here.
Okay, Seema, whenever you're ready, go ahead.
I think it's actually Jan who's going to go
Or
next.
Jan, okay, sorry. I got the schedule all mixed up this evening.
Hi,
Sorry.
everybody. I'm Jan Nachlinger, and I have been a volunteer for the GLORIA program for 16 years now. Can you all hear me okay?
We can hear you, Jan. We
Yes,
can't
Jan.
see you.
Okay, great. Next, Seema. So you heard the word silicates and dolomite earlier in the presentation, but I have separated the 2 substrates that the GLORIA target regions in the White Mountains are on as metavolcanics and the dolostones. White Mountain Peak itself is metavolcanic, and it is primarily composed of rock scree with a very little bare ground, whereas the dolostones that you see over on the right side with the bristlecone pines are mostly scree and bare ground with just a little bit of rock. Go ahead, Seema. So here's a closer view of the shoulder of White Mountain Peak on the left and the metavolcanic substrate compared to the dolostones in the Patriarch Grove area that are much lighter colored and primarily scree and bare ground. Next. And these are just a couple of the iconic plants that occur there. Then I'll discuss them in just a minute. So go ahead, Seema. The most ubiquitous plant in the White Mountains, as well as on all of the GLORIA peaks, is this Elamus elamoides, it's squirrel tail. It has a very broad elevation range, and it occurs in a great breadth of habitats. It actually some people actually have a hard time identifying it because it takes on so many different morphologies. It can be hairy, it could be hairless. The inflorescence sometimes is very tight, sometimes it's very open. So even though it is the most common plant, and it is the most common plant across all of the GLORIA target regions, it's very disconcerting to a lot of the botanists. Next. In the White Mountains, Polemonium chartaceum, or also called Partaceum, is the iconic high elevation flocks of the White Mountains. It also occurs in the Sweetwater Mountains to the north, and that's the only place that this particular species occurs. It is called Cretaceous because of the base of the leaves. In the lower center photograph, you can see some light colored at the base of these leaves, and that is that papery membrane that gives this plant its name. It also, on the right, you can see that it's quite glandular and hairy. And this is a characteristic that several of the alpine plants have, are these hairy glandular pubescence on the plant. Next. Cerastium beringianum is a member of the Caryophyll family, the pink family. It's at high elevations on the metavolcanics. It has 5 petals, although up in the upper left, you can see that it's bifid, so it looks like it has its notch, so it looks like there are 10 petals. And then over on the right, you can see that it's glandular and hairy as well, just like the polemonium. You see the seed capsules there that release the seed. And this is usually a very high elevation plant, but it will get down into the lower metavolcanics as well. Next. This plant is a member of the crucifer family. It has these white petals that look like a draba, but in fact it's A. Nelsonia eurycarpa. And even though it's got the nice white petals and very pubescent leaves that look like a draba, the fruit is very distinctive, and the common name is dagger pod. And that's also primarily just on the metavolcanics. Next. So Agropyron scribneri is also a very common grass up at high elevations. It has a synonym, Elymus scribneri, so it's very closely related to the squirrel tail. It is much more prostrate. It doesn't typically have pubescence like the squirrel tail does. And the inflorescence is quite a bit more elongated, so there's space on the between the florets compared to squirrel tail, but it's that prostrate habit that deciphers it for the most part. Over on the right is a very common high elevation bluegrass. It's Poakeckii, and it's just this cute little tufted grass that again is primarily on the metavolcanics. Next. Holzia algida, or alpine gold, is an anomalous plant in that it is tall in the alpine. You can see it right off of the shoulder here of White Mountain Peak. And this is kind of an unusual morphology for an alpine plant. It's a very glandular, hairy plant, and it's being studied right now, and these hairs may function in many ways. They possibly reduce water loss up in the alpine. They certainly could reflect sunlight. There's a possibility that they would capture prey for sources of nutrients, or those hairs may repel herbivores as well as possibly attract pollinators. So there's many possibilities on why it's such a tall and glandular hairy plant that it's going to be fun finding out what the researcher finds about this plant. Next. On the metavolcanics as well is this little member of the Montia family. It's Caloptridium umbulatum, or pussy toes, pussy paws, sorry. And it is anomalous in that it's a glabrous and succulent plant, which is not very typical for alpine morphologies. But this plant, like Elymus elymoides, has a very broad elevation zone, and it occurs in many habitat types. Way down low in elevation, it'll occur on barren land. So the main character of this plant is that its habitat are low competition sites. And so it does well up in the alpine, and it does well on barren areas down at very low elevations. It's got that broad distribution, just like the Elymus elymoides. Next. Oh, now this is plant candy for sure. This is Erigeron Vegas, a very high elevation plant on the metavolcanics. It does not occur down lower. It comes in these permutations of white, pink, and purple, and it's got the cutest little three-parted leaves with these very glandular hairs on them. So it's a very distinctive plant. Next. Androsaces septentrionalis is a member of the pink family. Higher up on the metavolcanics, it may be biennial, and as you can see on the right, it's a pretty good, robust looking plant. However, down lower, this plant does occur on the dolostones, and it is usually much smaller and an annual plant, and often only has a few flowers on it. So it depends on the year, and I happen to get a really good example of a very robust plant. It's about 1.5 inches tall. Next. A couple of more grasses on the metavolcanics are Festuca brachyphylla variety brevicolmus, a cute little fescue plant that is quite tufted and in full flower here. In the middle, there's bluegrass, Poa glauca variety rupicola, which is taller and more wiry than the Poa kekii you saw earlier, but it certainly isn't as big and robust as the Poa secunda that is much more prevalent throughout the White Mountains. The last plant here is a fern, Cystopteris fragilis, up in the metavolcanics, is typically hidden under the rocks down in the dark and not out here in the light. So I had to take this picture at lower elevations in order to give you an idea of what it looks like. But it is one of just a couple of ferns that we run into in the White Mountains. Next. Finally, we have a couple of ephemerals that showed up in the very good year 2011 on the metavolcanics. On the left is Erythranthes extorphii, a very tiny annual monkey flower. And on the right is Gymnostrus parvula, a member of the phlox family. And they're both very tiny, right next to Ellen's finger there, giving you an indication of just how many millimeters these plants are. We haven't had a good showing of annuals for many years now, unfortunately. Next. Phlox condensata is a prolific flower very early in the season. You have to be there just as the snow is melting in order to see all these beautiful flowers in great abundance. Normally, when we're sampling Gloria plots, the petals have already dried up and fallen off, and you just get the nice green turf of this plant. But this plant occurs on both the metavolcanics and the dolostones, and therefore is kind of the transitional plant that gets us down into lower elevations. Next. And here we are on the dolostones, lower in the Sheep Pass area with the metavolcanics way in the background up high on White Mountain Peak. This year was 2016, and it was an incredible display for Castanea nana, a member of the Phrymaceae, which is this plant is a hemiparasite and attaches to other plants to receive some of its nutrients. It comes in all sorts of permutations of the inflorescence in terms of its color, and sometimes it will hybridize with the lower elevation Castanea pilosa, a very abundant plant in some years. Next. Another member that's primarily on the dolostones, but it does get up on the metavolcanics of Barcroft Peak, is the White Mountains wild buckwheat, Eriogonum gracilipes. It's a very cute cespitous, densely tufted plant with a beautiful little pom-pom flower, and the leaves are often curled around in order to hold in that moisture with very, very nice soft hairs. It's really a prominent and very gorgeous plant. Next, a couple members of the Fabaceae or legume family. They both have purple flowers. However, they are quite a bit different. One, the Stragulus controfida variety Tegetarioides has a very prickly tips of the leaves, which is something that you do not want to kneel on, or you will notice it very dramatically, as opposed to Oxytropis perii, which has nice soft leaves, is not a bad thing to have to kneel on. The real difference between these two genera are in the flower characteristics, and unfortunately you can't see that from these images, but it is the shape of the keel, which is one of the specialized petals of the pea flower. So these are quite prevalent, mostly on the dolostones. They sometimes occur on the other rocks. Next. Another erigeron, a relative of the higher elevation Vegas, is Erigeron pygmaeus, a cute little tufted plant as well, but instead of having those three-lobed leaves, they're obovate and quite a bit less hairy. However, this plant does have those very purple involucral bracts beneath the rays of the flowers, just like Erigeron Vegas does, but just an absolutely charming bit of candy here. On the right is Draba oligosperma, a member of the crucifer family with its white petals and soft hairy fruits. However, the distinctive character of this particular plant are the hairs on the leaves in the upper right. Those are called pectinate hairs, and so they look like a comb, and pretty much have to have a good magnet to decipher that from the other Draba subumbellata that occurs up in the area as well. Okay, next. A member of the rose family is named for Jim Moorfield, the botanist in the White Mountains. Potentia moorfieldii is a very beautiful plant in the rose family. It's prostrate for the most part. It has the two-toned look to the leaves with pubescence on the backside of the leaves and a lot fewer hairs on the top side of the leaves, giving it that green look. Another character of this particular Potentia, and there are several of them up in the alpine, is that it has a very tiny styles, not really noticeable, perhaps on the rightmost flower on the bottom. You could see some of that. A member of the Linaceae is Linum lewisii. It's a tall plant that is very wandy and blows around in the alpine winds and will often lose those beautiful purple flowers, and then you either see it just in bud or in fruit, and you will miss those flowers just because it's sticking up in the wind, which is kind of unusual for some of these alpine plants. Okay, next slide. We are on the top of Sheep Pass, a group of us taking a look at the dolostones here, and we're finding all kinds of plant candy as a group. And in the lower right there, Seema was basically the one to find the two plants of our desire at this moment. They are both Townsenddias. Go ahead and forward, Seema. Townsenddia condensata is on the left. It's a much larger, relative speaking, plant with very soft hairs, and usually is solo. Whereas Townsendia leptodes is more clonal, and you'll see several smaller flowers together. And the leaves on this plant are less hairy and they're obovate. So they're pretty distinctive, but they're both really, really sweet plants. So I was only given 10 minutes. Sorry about that, but we are done now. Go ahead, Seema, and move forward. I wanted to thank the plants, the landscape, people, animals, the spirit, and White Mountains Research Center for everything that they're doing for us. Thank you very much.
Great. I'm just going to jump right in now that we know what all the beautiful plants are, thanks to Jan. So I'm going to talk a little bit about some of the science questions that we're currently addressing. And all of these questions are really related to understanding the long-term effects of climate change. And I'm going to start by talking about our work at small spatial scales, looking at climatic niches and functional traits of communities, both within and among peaks in the White Mountains. And then I'm going to zoom out a bit to talk about climate change impacts on species richness and composition of mountaintop communities in the 8 target regions that we monitor. And finally, I'll just briefly end by talking about some of the new and future projects that we're getting involved with, looking at climate change responses at continental and global scales. So before I keep going, just want to acknowledge the GLORIA Great Basin Board and co-authors of the work that I'm going to talk about today, and just a very special shout out to Kaleb Goff, who's the PhD student in my lab and is leading most of the work that I'm going to talk about right now. So starting with this first question, one way to think about how communities are responding to climate change is by thinking about the climatic niches in those communities. So if we look across an elevation gradient like the one shown here, we might expect to find a high abundance of warm, dry loving species at low elevations. These are depicted as different colored flowers here, and a high abundance of cool, wet loving species at high elevations. Over time, with climate change, we might expect increases in the abundance of these warm, dry, loving species. And this is a process that has been called thermophilization. And then another aspect of communities that could change across climatic gradients is functional traits. And functional traits are basically a way to describe the different ways that plants make a living. So there's been a bunch of work in biomes all over the world involving thousands of plant species that has shown that there are sort of these fundamental trade-offs between traits that allow plants to rapidly acquire resources, so things like having a tall stature, making thin, large, cheap leaves, and traits that allow plants to conserve resources in stressful environments. So these are things like having a shorter stature, making small, thick, expensive leaves. So we might expect communities to have more of these fast, resource acquisitive traits at low elevations and slow conservative traits at high elevations. And as climate changes over time, this could lead to shifts from these slow, expensive traits towards fast, cheap traits in plant communities. So we're currently addressing these hypotheses in the whites. I don't need to tell you more about that. You've heard all about it. So Jim described the downslope surveys. This is where we've surveyed plants on 5 peaks. We've established 8 to 14 transects downslope of each peak. Jim showed you the belt transects on all these peaks, but what I want to really highlight is that on each of these belt transects, we're basically collecting data on what plant species are present and what is the proportion of the 10 segments that are on that belt transect on which each plant species is found. And this is our measure of relative abundance. And we've got 3 to 4 surveys per each of these peaks that all happened from 2008 to 2024. So for all the species that have been documented on each peak, we quantified their climatic niches, and we did this using geographic data from herbarium specimens along with freely available climatic data. These efforts were led by Catherine and Sophie, who are 2 undergrads in the lab who Kaleb has mentored. And then we also collected functional trait data in the White Mountains, and Kaleb led this work with assistance from Malia and Sylvia. And then once we had the climatic niche data and the functional trait data, we used that to calculate abundance weighted means for each elevation transect on each peak. And so here in this figure, on the x-axis, we have elevation. And on the y, we have a measure of the average temperature niche of each of these alpine plant communities. And then each of these different colors represents one of the 5 peaks. And what you can see is that as elevation increases, the abundance of warm adapted species decreases. So this is just as we would expect. And then the black line here shows you this relationship across all peaks. And so the relationship is particularly strong when we look at this larger spatial scale of the elevation gradient across all peaks, but it varies in strength when we look within peaks. And just to show you a similar figure now for precipitation niche on the right here. So what this is showing is, as we might have expected, that we find wetter precipitation niches in communities that are at higher elevations. So now I'm showing you a similar graph for the average maximum plant height. This is just a measure of the plant's overall stature in each of these communities. And as you might expect, when you go higher in elevation, we see species with shorter heights on average. And this makes sense given, as Jan showed you, at the highest elevations, we see all the cute, short-statured cushion plants, right? So this is overall consistent with our hypothesis that there would be these more resource acquisitive, fast traits at low elevations and more conservative, slow traits at high elevations. Another trait that we looked at is specific leaf area. This is a measure of high values of specific leaf area tell us sort of more or less that leaves are relatively large and thin. Low values correspond to leaves that are relatively thick. And so what we found here was pretty surprising. We found that actually as elevation increased, specific leaf area increase. So we saw at the highest elevations, communities tended to have leaves that are thinner and larger relative to low elevations. And what we suspect is that this increased drought stress at low elevations favored more resource conservative leaves. So the next thing that we did is we looked at how these climatic niches and functional traits are changing in these communities over time with climate change. So this graph shows you just one peak, White Mountain Peak. On the x-axis, we have year, and on the y, again, we have that community-weighted temperature niche. And so, and then the transects are colored by elevation. And what you can see is that there's really not much change in the community-weighted temperature niche on White Mountain Peak. And when we look at maximum plant height, we found the same thing. And when we looked at this, all the climatic niche variables and all the functional traits that we examined on all 5 peaks, we found really similar patterns where we're just not seeing changes in these communities in traits or climatic niches over time. So to kind of sum this part of the talk up, we found that the abundance of cool, wet-loving species increased with elevation, particularly across peaks, but it didn't decrease over time with climate change as we originally expected. When it comes to functional traits, we found that communities tended to have shorter statures as elevation increased, but contrary to our expectation, they also had these sort of thinner, cheaper leaves. And we think that there's sort of these contrasting effects of temperature stress at high elevations and drought stress at low elevations in the White Mountains, and that this might have decoupled these functional traits and sort of broken what has been referred to as the plant economic spectrum, these trait correlations that have been ubiquitous across a bunch of different plant species. So we found this really interesting. Similar to climatic niches, there really wasn't much evidence for changes in functional traits over time in these communities. So for that first question, we were focusing on 5 peaks within our 2 target regions in the White Mountains. But for the second part, we're going to sort of zoom out a bit to ask questions about how species richness and community composition are changing over time with climate change across all of the 8 target regions that we monitor through GLORIA Great Basin. So this has already mostly been covered, but alpine plant communities are a really nice system for testing hypotheses about responses to climate change, given their sensitivity to climatic conditions. But there's this sort of tension where on one hand, mountains have sometimes been described as these escalators to extinction, where species are shifting their ranges upslope to track their climatic niches until eventually they get to the top and run out of places to go. But on the other hand, mountain landscapes are topographically heterogeneous, so there could be lots of little suitable pockets of microclimate that buffer plants from changing climate. And this could lead to overall community resilience. So one big question that we haven't quite answered is, collectively, how will range shifts of individual species affect community assembly with climate change? And so we can think about 3 time lags to range shifts, which are shown here along an elevation gradient in an alpine region. So first, there could be lags in dispersal rates of species from low elevations to suitable climates upslope. Second, there could be lags in the establishment of these species once they disperse to high elevations. And then finally, there might be lags in extinction of the resident species on the very tops of these alpine summits. And so these different kinds of lags can have really important effects on the numbers of species that we see on mountaintops, as Brian was talking about at the beginning of our talk, and also the identities of these species, who's going to be the winners and the losers over time with climate change. So this graph shows you an example of one climate variable, mean annual temperature, from 1980 to 2022 in each of our 8 target regions. And the gray shading just corresponds to historic temperatures prior to the start of the study period. And the study period runs from 2004 to 2022. So you can see that it has gotten warmer just over the study period of over 19 years. The mean annual temperature has increased by a little over 1 degree Celsius. There's not really a clear trend in precipitation, just lots of whiplash and variation across years. So Jim talked about a lot of the details of how we set up our plots and the different spatial scales, but for the purposes of what I'm talking about right now, we're just going to be focused. So we have our 8 target regions. We have 4 peaks, 3 to 4 peaks arranged along an elevation gradient. In White Mountains, the tallest peak would be White Mountain Peak, of course. And on each, and then we're just surveying the top 10 vertical meters of each of these peaks. So we're lumping all the smaller aggregated data together to look at what species were present on these top 10 vertical meters of each peak, what was their categorical abundance. And so we then could use that data to quantify species richness and composition for each peak and each year. Sip of water. So I'm showing you what that looks like in one of our target regions, the White Mountains, quartzites or silicates, also called metavolcanic, as Jan was calling them. So here we have on the x-axis year and on the y-axis species richness. And each of these points is a given peak. The elevation range of the peak is shown by different shapes. So these triangles on the bottom show you the highest elevation, in this case, White Mountain Peak, and the squares show you the lowest elevation peak. So something that you might notice is that species richness, no matter what time point you look at, is lowest at the highest elevation peaks and highest at the lowest elevation peak. So this is kind of follows what you might expect. But then when you look at how species richness changes over time, although you might see that maybe if you squint, there's a slight trend of species richness increasing, the slope in this figure is not statistically different from zero, and these positive trends are really quite weak. And so basically what we found for this target region is that species richness did not change much over time on these peaks. When we look at this, the peaks of all of the other target regions also, we find the same result where surprisingly, despite these increases in mean annual air temperature that I showed you, species richness has remained relatively stable over time. So this really begs the question of, why is it that species richness isn't changing? Is it that we're just seeing the exact same species on each peak, survey after survey, or are the gains of new species and the losses of resident species sort of balancing each other out? And so to look at this, we quantified proportional turnover. And this figure, each of these small gray dots represents the proportional turnover between two consecutive surveys on a given peak. And what I want you to get from this figure is that on average, turnover was about 22%. But in some cases, you can see it was even higher than that, sometimes over 40%. And so what this suggests is that although species richness remains stable over time, there must be sort of a balance of species gains and losses that's underlying that stable richness. And just as an aside, many of the species being gained and lost are relatively rare forbs, and some are annual species. But it really seems to be mostly chance colonization and extinction events that are driving this turnover rather than some clear directional signal of cold adapted mountaintop species being lost and warm adapted lower elevation species being gained due to climate change. So just to summarize, despite recent increases in temperature, we didn't find much change in species richness over time in any of the mountaintops in our 8 target regions. Yet we did find substantial changes in composition driven by sort of a balance of species gains and species losses, but no clear evidence of directional community shifts due to climate change yet. So collectively, these 2 parts of my talk, what we've found so far is that we've established this really nice baseline data set that shows predictable shifts in community climatic niches, functional traits, and species richness with elevation. But those shifts that we're observing across spatial gradients and climate are not yet happening over time with climate change in the regions that we study. And so what we really need to do is a little bit more work to understand the extent to which this observed stability over time is due to these different kinds of lags that I talked about in dispersal, establishment, and/or extinction. And another thing that we are really excited to do is expand in geographic scale to better understand how these responses might vary across regions that are experiencing really different directions and magnitudes of climate change. And this is exactly what we're starting to do now. So in close collaboration with the National Park Service Inventory and Monitoring Program, we are starting a continental analysis to assess alpine species vulnerability and to look for summit characteristics that predict similar responses to climate change. And we're really lucky to have Megan Oldfather from USGS as the leader of this work. I'm sure many of you know Megan. She's a former co-director of GLORIA Great Basin and did her PhD research in the White Mountains with IVSIA. And so thanks to Megan, nearly 30 target regions all across North America have already agreed to participate in this continental synthesis. And thanks to the generous I&M funding from the national park, Kaleb Goff is going to be helping lead some of these efforts through a PhD research assistantship and fellowship, graduate fellowship. And I also want to really highlight the major contributions from Abby Volk, Brian Smithers, and Kristen Legg, who have been really closely involved in fundamental in making this happen. Just in the interest of time, I'm not going to spend a lot of time on these questions, but some of the focal questions for the synthesis that we're excited to tackle are asking questions about assessing vulnerabilities of management relevant species, questions about alpine greening, whether that's happening on some summits, and whether we're seeing species shifting their ranges across aspect or elevation. And then finally, we're really, really excited to be able to contribute our data from the White Mountains and the other GLORIA Great Basin target regions to a global synthesis, which is going to be led by Harold Pauli and several other European scientists, thanks to funding that they receive from the Austrian Research Fund. And so some of the hypotheses that they're interested in examining are things like whether regional velocities of climate change are driving changes in species richness and composition, whether combinations of warming and drought are accelerating species losses in the alpine, and finally, testing whether there have been losses and or declines in small range species and gains in large range species, which could lead to the homogenization of mountaintop communities at various spatial scales. And so I just want to end my part of the talk by saying thanks to all the support that we've received from WMRC, all the volunteers who have helped us collect data, many federal workers who have made really huge contributions, and various funding agencies, many of which are federal funding agencies. What started as one of GLORIA's international master sites in the early 2000s is now in the position to contribute over 20 years of data from 8 target regions to this global synthesis. So we're super excited about that. Okay, thank you. And I'm going to pass it on to Kaleb.
Hi, everybody. Kaleb here. I have the privilege of sort of taking an end run around this talk and showing some gratitude and really by now you've heard a lot about GLORIA's history and about field methods and science. And I think by now, Jim even said this unpromptedly, right? That you realize that the power that goes into all this is the power of people. And something that's really striking to me. Here's just 3 photos from the 2024 GLORIA Great Basin field campaigns. But I'm just struck by just the quality and the beauty of the people that come and join us out in the mountains. So just a couple of quick words about that and a couple of quick words of thanks. Seema, you can go ahead and go to the next slide. I have a bunch of roles being part of GLORIA Great Basin, but one of them is to be GLORIA Great Basin field coordinator. And one of those responsibilities is to sort of take the time that we spend together in the mountains and make that go beyond or sort of revel in the ways that goes beyond just getting the surveys done and just getting the data and just bearing witness to the place and just identifying the plants. So a little bit of hail frolicking. I know Jim cautioned us against playing in the thunderstorms, but we're close to the safety of the field station, some nature journaling, some song and story in the evening, glow-in-the-dark bocce ball, which came around from before my time, working with GLORIA Great Basin. There's a picture in the lower right of me trying to fit through a cave squeeze before we went on a cave tour in Great Basin National Park. Next slide, Seema. And then another project that we've been working on for the last 3 years that have been filling me with a lot of light and life is the GLORIA Great Basin Peak Opportunities Fellowship. And this is a fellowship that seeks to bring more diverse perspectives into mountain science. And what it does is it funds undergraduate, post-baccalaureate, and graduate researchers to come to the White Mountains and to survey peaks in the White Mountains, to stay at Crooked Creek. It provides them with a stipend so they can get the field gear that they need to be safe and comfortable out there. And I think, most of all, puts them in the Crooked Creek dining hall to talk to all the amazing, brilliant, and beautiful people that come up and join for these surveys. And right now, the application period for this is open. So if you're an undergraduate, post-baccalaureate, or graduate student, go over to our website and check this out. Applications close on April 1st. So we're really excited about this. Next slide, Seema. And then, of course, one of my wonderful duties here is to try to get people to come out to the surveys, and luckily that's not a big challenge. Lots and lots of people. You can imagine over the past 20 years, hundreds and hundreds of people have joined us on these surveys. This year, we are going out for the 5th survey, the 4th resurvey, 4 silicate summits in the White Mountains. So that's going to be, the dates are there, and then 4 more summits in the Dunderberg Peak Yosemite region. Again, also, yeah, both of these target regions started in 2004. So this is a really special one. So 20 years later, coming back around to this place. So feel free to go to our website and you can get on our mailing list. Our sort of rhythm for the year is for me to send out an email fairly soon here in the next week or two, soliciting interest, and then follow back up and get a little bit more firm commitments to these dates. Then I get everyone ready and we join each other in the field. So excited about that. And then just a soft reminder to say that you can also, join and be part of this cause by donating. A lot of the funding for GLORIA Great Basin, or some of the funding from GLORIA Great Basin, comes from people who are willing to give us some of their energetic currency and some of their monetary currency. So we really appreciate that. Next slide, Seema. I'll just lastly say that this project really represents this deep well of gratitude. You've heard that already a bit, but I'll just say, right, hundreds and hundreds of people have taken part in this work and sort of contributed their beautiful gifts. We've collaborated with all these amazing government workers and land management organizations, got funding from all these sources. I won't read through all of these, but really, this is just such an important collective effort, and I'm just personally so grateful to be part of it, and I think we are collectively grateful to be able to share just a little taste of it with you this evening. So I'll wrap up by saying thanks for your thoughts and attention. Go ahead and advance. Thank you. I think we have a couple minutes to hear your questions, and just end with a few words from Gary Snyder. Stay together, learn the flowers, and go light. We're able to be in these beautiful, amazing places and to bear witness to them, and to make something beautiful together. So thanks so much for having us this evening. And you can reach out to us by emailing. You'll email me if you email coordinator@gloriagreatbasin.org, and happy to chat, and can also get you to talking with some of the other people who are involved in GLORIA. So thanks so much.
Thank you, Kaleb. Thank you, Seema. Thank you, Jan, Jim, and Brian. So we are getting a little close on time, and for our viewers in the future and YouTube, I just wanted to read some of the comments in the chat, so then folks have a general idea of about some of the questions asked by the audience. So Alex had asked if you guys had collected any plants for voucher specimen, and then Brian had answered that in the chat, that you guys very rarely collect plant materials, and the data that you do collect are monitoring data, and so you guys are noting its presence and abundance. On occasion, you do need to collect plant material for identification purposes, which then you do send outside of the plot, and you have them placed into the herbarium at UNR Reno for future use, excuse me. And then there's another comment too from Seema saying that she forgot to say that she's very interested in recruiting a PhD student to work on GLORIA related questions. Please spread the word. Yeah, and then if anyone has any questions from the audience, you're more than welcome to type the question in the chat or turn on your video and raise your hand. We do have some time left. I have some short announcements to make afterwards. I also do read praise from the chat. Great presentation. Thanks, everybody. Wonderful presentation. Also, thank you again. This is our largest group of presenters virtually. Virtually, Brian. Yeah, and then I saw too that there was a citation in press for your paper, Kaleb. So we'll please send that so we can database it. We can, cite it in our database. And as you know, you're all welcome. All welcome to share project summaries on our website too. We do get a lot of traffic on our website, especially it's our 75th anniversary too. And so we have Gloria. And would this be your 22nd year? 21st. Okay.
Gloria turns 21 this year.
Oh,
No
okay.
innuendo
Behave yourselves,
intended.
please. So I guess I will make a quick announcement. Our next talk is next Thursday, March 13th, and it's in person. So we're going to start in-person talks. And that will be at the Owens Valley Station in Bishop on 3000 East Line Street. There is more information on our website, wmrc.edu. The topic of this talk will be 100 Years of Alice Piper with Big Pine Paiute tribal member and Akamai Cultural Group Director Sage Romero. Sage will be discussing the life and legacy of his late family member, Alice Piper. The Piper versus Big Pine case set precedent for the Brown versus Board of Education case during the civil rights movement. So big things happened in the Owens Valley. And yet again, you can visit our website for that schedule. I also wanted to thank you guys too. I know that you're presenting from at least 3 different time zones right now, and that it's getting late. So I really want to thank you for doing that, and that, hosting virtual talks really allows for more participation from researchers and the audience. So thank you. Does anyone have any questions? It's pretty quiet. Do you guys have any more final thoughts? I also wanted to acknowledge Mr. Cohen. Him and his late wife Valerie were also instrumental in attendance and participation in the GLORIA surveys. Valerie was a very well-known artist in the Great Basin area. And so we just want to acknowledge hers and Michael's contribution as well from WMRC and, GLORIA. So thank you.
Hi, this is Glenn McDonald. I'm the faculty
Hi,
director. I'm the faculty director at White Mountain Research Center. I learned about GLORIA when it came to UCLA in the Institute of the Environment. Tony Orm was the director of White Mountains then. He was a big fan of GLORIA. He just had to tell me a little bit about it, and I was all in. And I just want to say that that was a fantastic presentation. Your work is so important because getting longitudinal data like this is very, very difficult. And we are proud. We are proud to do what we can to help with GLORIA, and we hope this goes on for a very, very long time. So thank you all very much. You really, really make us proud. That's enough.
Thanks, Glenn. Yeah, and I was about to say kind of something similar, that the only reason this has happened for as long as it has is because of our partnership with White Mountain Research Center and the creativity, we'll call it, that you all have used to make it so that it works, that it works for both of us. So thank you. Yeah, it's a great partnership, and we it's a great symbiosis, I feel like.
Thank you, Glenn. Thank you, everyone, for coming. And thank you also to other WMRC staff and former partners that have attended tonight. Also, this talk is being recorded. I will get it posted probably next week to our YouTube channel. The handle is in the chat. If not, if you just Google search it, it will come up. All of our lectures since 2021 have been recorded and put on there for your viewing pleasure and expansion of the mind. So with that, I'm going to end for this evening, and I will probably follow up with you guys, Gloria, in probably email format later on, either tomorrow or earlier next week. Okay. All righty. Thank you so much, everyone, and have a good evening. Okay. Okay. Good night.
Thank
Thank you all.
you.